All set when you're set.
All right, good morning. This is Jason Kolbert. I'm the senior analyst at Boral Capital. I cover Clean Therapeutics, which is working on a very interesting strategy around Lou Gehrig's disease or ALS. Today, we're talking with the CEO of the company, Etherington. Hope I got that right. And
Perfect.
Rob is going to tell us a little bit about the company, a little about the history of the molecule, a little bit about the history, clinical history, and then looking forward into the future about what it's going to take to get this product approved. So, Rob, can you give us an introduction to who you are, what attracted you to the company, and what is nanoparticle gold all about?
Certainly, Jason. First, thanks for the invitation and greetings to everybody out there. It's a pleasure to talk to you today. So I spent 9 years at Pfizer, kind of classic big pharma experience, working on, among other drugs, Lipitor. So these are recipes of chemistry and solving cholesterol problems. And then I went to a small little company based in Basel, Switzerland, to spend my time in rare orphan diseases that at the time were uniformly fatal. This is a company called Actelion. It became the largest biotech in Europe whilst I was there, and we focused on pulmonary arterial hypertension, which is high blood pressure in the lungs. And before the drug that we developed was approved, patients had to have a heart-lung transplant or they passed away. So here we are in the case of ALS and neurodegenerative diseases generally. And so while I was at this last company, where I stayed 13 years, Actelion, I was contacted by some individuals who said we're thinking of something super novel, which is a new therapeutic area. So if there's small molecules, recipes of chemistry, that we are not that. If there's biologics, we are not that either. What we are is an intersection of physics and material science into biology, which is the idea of using a gold nanoparticle at at nanoscale. So this is small enough to cross the blood-brain barrier and to go inside the mitochondria and to drive metabolic energetic change, which is critical because you and I and the entire human family, we slow down as we get older. And if we couple that with a neurodegenerative disease that is attacking the very essence of what gives us life and causing tremendous, devastating compromise in function, as ALS does indeed invoke, then we've got massive problems. And so I thought it would be, back to your point about the origin story, I thought it would be pretty extraordinary to see if we could come and do something novel and unique. Now, the tricky thing is that there's never breakthroughs always, as Stephen Covey, the famed author, used to say, are always break with convention. And the idea that a nano suspension that somebody drinks orally could come into the bloodstream and cross the blood-brain barrier to drive nicotinamide adenine dinucleotide or adenosine triphosphate change in the mitochondria of the central nervous system is a pretty big idea. And how one would do that with a gold nanoparticle is also pretty extraordinary. So there's been a lot of skeptics in the last 12 years we've been
I
doing
just
this.
want to translate a little bit about what you just said, which is the energy function of the cell, or which is comes down at a molecular level to ADP, and some of us might remember the Krebs cycle from our high school biology. You're talking about basically cellular energy, and I think you're going to relate it now to this concept of nanoparticle gold, which is not the shiny gold bullion bars that we see. It's actually a microscopically small liquid formulation, so small that it can cross the blood-brain barrier.
Yeah, that's correct. In fact, yeah, it's very important we distinguish. We're not talking really about gold in a wedding ring, but we are in a way. So let's just go back to this. So there's been a long understanding of the utility of therapeutic elemental metals in science. If we went and looked online now, we would learn about colloidal silvers for an antibacterial or antiviral preparation. For decades, a kind of a key aspect of chemotherapy regimens in cancer is a drug called cisplatin. The platin piece is about platinum and the chemistry level. Zinc we take to prevent colds, and gold was injected into our grandmother's rheumatoid arthritic joints for decades. Kind of from the 50s to the 80s, gold salt injections were standard of care in rheumatoid arthritis patients. And so we had this idea that if we could use at nanoscale, and again, this is super small, a catalyst that could drive energy response into a failing or compromised neuron, this could matter quite immensely. And so it's a different non-chemical approach. It's an intersection of material science and physics into biology because this gets a little wonky, and so we probably should go too deep here. But gold on the elemental table is missing an electron in its s orbital, and so by consequence, it's a highly reactive catalyst. And so to serve as a catalyst in a failing neurological system, and I mean failing because in ALS, that's what's happening. For a number of reasons, the body is attacking itself. And the consequence of that is my neuron is dying. And the consequence of that is I'm no longer able to send signals electrically to my neurons to execute muscular function. And the consequence of that is I start being able to have compromise in the way I speak, the way I breathe, the way I move, the way I walk, the way I feed myself, and so forth.
Terrible disease.
Yeah, terrible, devastating, devastating in its consequence. And let's turn to the disease for a second. So ALS famously was given a nickname, Lou Gehrig's disease, because of the famed baseball player in the 30s that was diagnosed with ALS, struck really down at the prime of his life, as so many ALS patients are. And so years we called this Lou Gehrig's disease. And since the Ice Bucket Challenge, we've really tried to move back to amyotrophic lateral sclerosis, its true name. And rest of world, it's called motor neuron disease. But in any event, it was decades before there was any therapy to be prescribed. The first drug that was effectively scientifically proven to help is a drug called riluzole that was approved in 1995. So that is 30 years ago. That drug still is standard of care therapy. So,
And marginal efficacy.
and marginal efficacy. Everybody's on it. You and I would be on it too if we had ALS. Everybody's on it. It's been proven definitively worldwide to have marginal, but indeed efficacy. And since then, there's been very little progress, sadly. There was another drug approved 10 years later, and there was another drug approved for a very rare form of ALS by the pharmaceutical company Biogen. But other than that, patients have so little. So
And
very,
the
very,
current drugs, the hope is to change the course of the disease, the slope, but they're not there's no such thing as a cure at this point.
there's,
And it's a balance of whatever the limited efficacy is, and it's limited, versus the adverse events. And I wondered if you could just briefly talk about what is the adverse event profile when we talk about nanoparticle gold, because I think that's one of the unique characteristics.
Yes, indeed. Now, one of the reasons we call the company Clean, in fact, is because we theorized at the beginning of this that we would, in fact, have a remarkably safe asset. And that is indeed the case, gratefully, knock on wood. We have over 1,000 participant years of experience now to our CMAO8, what we call our drug. We have long-term treatment duration at 6 years and counting. People with ALS that have had our drug, taking it chronically every day for up to 6+ years. And I know some of these patients. I've met them. I was just with one of them actually in a Zoom call a week, Friday, in fact. So this is somebody that started our drug in 2019, and to do that in ALS is a remarkable thing. Now we have some side effects. We have some headache and some nausea that occasionally occur, but these are mild to moderate in severity. They're also transient, which means they go away. And there's also no temporal association of increasing incidence based on exposure, which means that the longer you're on our drug, you're not getting more side effects, you're getting less. And critically, what we count in our world is what are called serious adverse events that are related to drug. And always pharmaceutical companies and their drugs have some of these. We have, kind of remarkably, not a single related serious adverse event that has been related to our drug from a clinician in clinical studies in any of our programs, not in our Parkinson's, multiple sclerosis, or ALS work.
Well,
So this
so as a biotech analyst, I'll tell you, I see drugs fail to be commercialized for one or two reasons. And the one of the most predominant ones is adverse events, right? When you have to weigh benefits versus risk. And what I hear you saying is that you've already established a very favorable risk profile, so that the challenge now is to be able to effectively show what is the benefit. Is
that is
that fair?
correct. That is correct. Yes, the agency. So, as a nanotherapeutic, they put us through our paces. About 10 years ago, we started showing them a bunch of animal work, which is typical in our world, to really provide deep evidence that a nanoparticle was not hurting folks, especially one that was taken orally. Because as I drink it, and that, by the way, is how one takes our drug. One drinks it every morning if one is in a clinical study. Just as an aside, it's as if I was drinking a glass of water. That's the best metaphor I can give people. In fact, all of the placebo data that we have, so in double-blind placebo-controlled studies, of which Clene has concluded five of these, there's an active arm where patients drink CNM-Au8, and there's a placebo arm where they drink, in our case, their placebo is purple-colored water. Purple colored water is our placebo because our drug CNM-Au8 has no taste. It has no taste at all. In fact, it tastes like, as patients tell me, room temperature water. And so that's why the placebo is indeed purple colored water. But in any event, back to the point, we had to give these animals super high doses. We had to do a bunch of work to prove safety. And then ever since then, the FDA has got increasingly more comfortable as the clinical data rolls in from all these patients treated. So, how many now? Well, we have about more than 200 that have been treated in double-blind placebo-controlled studies. In fact, it might be 2 to 250. We have more than 500 that have been treated in compassionate use expanded access protocols.
Wow.
So EAP is a specific United States designation of a clinical protocol that the FDA allows for expanded access to an experimental therapy. It's called EAP. And we have four EAPs that have been approved by the US FDA for people in three in ALS and one in multiple sclerosis for individuals to try our drug in compassionate use. One of which is the NIH-sponsored EAP, the largest of our programs. And the National Institute of Health gave Clene a very large grant from this extraordinary pile of money that Congress authorized for the development of ALS science called the Act for ALS. And Clene is super grateful for that money. And that has, in fact, taken us to where we are today with an FDA discussion upcoming in the first quarter, in fact.
Good, great, thank you. And you mentioned briefly clinical data. I know that there are two prior trials, Rescue and Healy. Can you talk a little bit about what was Rescue? How many patients? What did you learn? How is that related to Healy? And how does that form the foundation of your thinking as you look at the next clinical step?
Sure. So Rescue was our first program that commenced. It was a study done relatively small, 45 participants in Australia. It was funded by a charity called the Fight Motor Neuron Disease Association, which was founded by a series of athletes that unfortunately had ALS. ALS is unfortunately more common in athletes and in veterans of armed services. Not exactly certain. There's a number of theories as to why this is the case, but in any event, these athletes funded this work. We got a chunk of funding for rescue. Rescue was in early patients. In fact, the average diagnosis were individuals that were only 4 months diagnosed, so pretty early in their disease progress. Our primary endpoint was an innovative one that was called MUNIX. It stands for Motor Unit Index. And we looked specifically at the motor neurons in the hand, arm, and leg to understand how they were able to retain and maintain function. So, on the primary endpoint, looking at all subjects, which includes limb onset and bulbar onset individuals, and just a little bit of understanding for your audience. Bulbar onset ALS is when it originates, the disease ALS originates in the bulbar region, which is in my bulbar region of my brain. And patients present with breathing challenges, eating challenges, chewing challenges, and speaking challenges. So we enrolled both bulbar and limb onset, but we were only evaluating limb. And so we missed the primary endpoint. We were massively underpowered. But what we saw is that we saw a decreased time to clinical worsening. And clinical worsening in ALS is defined as whether I can stop breathing, whether I need help with respiratory support, that is, whether I have to stop eating because I can't swallow food and I need to go on a gastrostomy tube, and whether or not the ultimate clinical worsening would be passing away and death. And we
I
saw,
just want to interrupt one thing you said, which is that you missed a primary endpoint. And I think in some ways it's kind of an insane statement because at 45 very early onset patients, other than 100% efficacious drug, it's almost impossible to hit a primary endpoint. So it's really the search for the right patient, the right dose, and determination of the signal, right? The informative design, if you will. Is that fair?
that is very fair. And there was three pioneering things about this study. A, Munich's had never been done. This notion of evaluating motor neurons in a phase 2 clinical study of ALS. We were pioneering that. We are pioneering in early disease, and we are pioneering in including bulbar individuals, that is, with this bulbar region compromise, in a disease where we were looking only at limbs as an endpoint.
Yeah, I mean,
So
very, very challenging and on every level
you
so the
learn a
fact that you
lot.
could show a signal is kind of amazing, right? That's why I want to twist that around. I don't really see it as a failure. I see it as in 45 patients, you were able to show a signal and very unique patients, right? Very unique clinical
Well,
parameters.
well, to the point, which I appreciate the point you're making is that if we would have looked only at limb patients and not included the bulbars, which is hard to do, patients that have bulbar, they don't want to be excluded from clinical
Of
studies.
course not. Right.
But if we'd have only looked at limb only in this case, then the data in a post hoc analysis, which means to your viewers after the fact, we've evaluated the data. We probably would have hit the primary endpoint if we'd have
Yeah.
done that, but we
Okay.
didn't. So we learned a lot, but we also saw two other things happening, which have informed us ever since. We've learned that in this study that we saw some progression of biomarkers that were sending us, even though super small, some very interesting, intriguing pieces. We saw in the open label extension survival benefit. We saw in the open label extension preservation of function. And so that really gave rise to the key next study that you just referenced, which is the Healy ALS platform study. So, a little background on that. Dr. Merit Sudovich and her colleagues and multiple centers across the country, starting in our case with more than 50, and now it's expanded even beyond that, came up with this extraordinarily clever idea to see if we could take a number of assets with a platform study and progress these assets down in ALS. And so, what a platform study means is that you recruit individuals with the same inclusion and same exclusion criteria. They're all on the same protocol. You're on your drug, but you're sharing placebo across a few arms, and you're all evaluating your drug on the same endpoint in the same protocol. So it's a really an opportunity to be efficient in clinical design, and we were one of the first drugs chosen. The Healy ALS platform study has now completed 8 different regimens. All of these were 6-month studies to date, and that's going to be relevant in a minute. I'll talk about why that time is tricky. And all 8 of them have, in fact, missed the primary endpoint of ALS-FRS. But to your point of clinical design, Clean saw some very interesting things. We saw a secondary achievement of survival. We saw a pre-specified benefit of the exploratory clinical worsening, which we also saw in rescue. And we saw, very critically, a reduction in the pre-specified design of a biomarker called neurofilament light. And I'll come back to what that is in just a second. But these 3 things, clinical worsening, survival, critical endpoint, and biomarker, that then continued in the open label extension. We saw more evidence of survival, more evidence of biomarker change. That then led us on a discussion that we're on presently with the US FDA.
Gotcha. Well, I've learned one thing, and that's that the FDA loves if they can correlate a biomarker, right? That becomes a very significant clinical parameter for them. Survival is always the gold standard, but I don't know if that's really true in ALS, and it's always been ALS FR. So obviously you have a lot of things that you must be thinking about. So take us a little bit about what's the next step here. And obviously it sounds like you've been dialoguing a lot with regulators. Talk about your regulatory interaction as well.
Let me talk about 2 things that I just introduced that you highlighted, and then we'll jump into the regulatory piece.
Sure.
So on survival, yes, overall survival, totality of evidence, whether or not people live or die is a super obviously definitive endpoint. And if we'd have designed our study with survival, we would have never thought of that at the time. This benefit that we saw of survival in both rescue in the open label and in the double-blind period, which was much larger, by the way. The Healy study was 4 times larger than rescue. So we had the powering that we needed to see this benefit. This was unexpected, to be candid. And ever since then, survival has been a key piece for Clean. But there's not a statute that the US FDA has in their regulatory guidance on survival alone. There is, though, with what's called a surrogate endpoint. And a surrogate endpoint is defined, among others, in the case of ALS, as a biomarker. And so let me just highlight, I mentioned about 5 minutes ago, neurofilament light. So what is neurofilament light? So this is a structural protein of the neuronal axon. So my neuron that is enabling this choreography of life, of movement, is very critical. And inside it is a structural protein. And neurofilament light is released as a bit of evidence of a debris field as the neuron dies. It's a cytoskeletal protein remainder that is resulting from the axons being damaged and degenerating in ALS. And it enters the bloodstream, it enters the cerebral spinal fluid, and we can measure it.
All right.
So
It's the
elevated.
equivalent of, say, an ALT to a liver.
Yes. Or even, even a weaker example than yours, but might be familiar for many of your listeners, cholesterol. If I have high cholesterol levels, I should be worried about atherosclerotic disease. Or, I'm now a 59-year-old man, so I need to worry about prostate markers, my prostate specific antigen to make sure my prostate's being a good boy. Similarly, if I have high neurofilament levels, I need to worry because elevated neurofilament levels strongly correlate with faster ALS progression and shorter survival. So we saw neurofilament light chain um, improvement relative to placebo that was a statistically significant way in Healy. And then we just announced last week, in fact, another biomarker called GFAP, stands for glial fibrillary acidic protein. This is similarly a structural protein, but not in the neuron here, in the astrocyte. And the astrocyte is crucial for supporting the neuron. And if I've got high GFAP levels also in my bloodstream, that reflects a very harmful inflammatory and degenerative process, which is evidence of the motor neuron loss in ALS. So we don't want elevated levels of either of these in our bloodstream. They're both very relevant ALS disease relevant biomarkers. So what we're doing now is the FDA does have a path called accelerated approval, which is a term that harkens back decades. It really started to become, if you will, in the AIDS era, where people were dying, and we needed a little bit of an accelerated approval path based around a surrogate endpoint that was different than the long primary endpoint in a clinical study classically. And so the agency has, to your point, we've met with them a number of times. November, June, August, September, and then we are now planning to meet with them in the first quarter. So 5 times in, what's that, 13 months?
Yeah,
Uh,
that's amazing.
yeah, actually, I'm a little exaggerative there. Five
I can
times in,
only think of a handful of companies and times in my career that have met with the FDA that many times, basically within a year. So that kind of suggests to me that they have an interest.
well, we are super grateful for their collaboration, their partnership. So all of these meetings are with the Division of Neurology, and we really applaud Dr. Teresa Baracchio and in particular the ALS clinical team of Division of Neurology 1, Emily Freelich. They have, it's the ALS is a difficult disease. And to look at this somewhat with some regulatory flexibility, we are super grateful for that. And by that, I reference a very interesting thing that happened in the late part of last year, so basically 12 months ago, where the agency told us in minutes that we got back in December, so literally 12 months ago from now, that we could look at the $45 million NIH-funded expanded access protocol for extra data that would give them greater confidence of the neurofilament light biomarker response. And so what that really references is the agency didn't want to just lean on one single study, which is the Healy data, and that response in neurofilament light alone. They wanted to look at additional data. So they carved out 3 paths for us, one of which is this NIH-sponsored EAP, to look at that data where neurofilament light is captured in a completely different patient population than was studied clinically. And just to make that point, the Healy study looked at folks that were about 6 to 12 months diagnosed. A classical expanded access compassionate use program is looking at folks, uh, ALS, people with ALS all over the spectrum, from early disease to super late disease. Anybody that basically wanted our drug, if they came into the expanded access protocol and got, you know, requested the drug through the clinicians, we gave it to them. So it was a very broad, uh, different patient population than has ever been studied. But in those patients, we were looking at neurofilament light regularly. And so the FDA said, let's use that data and see if you see a concordant response. Let's also look at the placebo individuals from the Healy study who converted to active drug. Did you see data there? And then third, let's see if you can find other biomarkers that are disease relevant besides just neurofilament light. And the agency had us organize a statistical analysis path forward with a protocol that we developed with them. And then that is the data that we've just presented to the markets last week, and that we're presenting to the FDA in a full dossier that we'll be submitting to them this month for a discussion that will happen in the mid part of the first quarter.
If the discussion in Q1 goes well, what does the timeline look like, and can you opine on what the clinical trial might look like in terms of a primary, a secondary, and what the powering you're thinking would be? Because that'll give us a good feel for how long the trial will take and what it would cost.
Sure. In fact, you've just raised a very important point that we've not yet talked about. So let's address that now. And that is that the design of accelerated approval presumes and indeed is conditioned upon that the company does a confirmatory phase 3 study. So, what the agency is effectively doing is saying, look, we recognize there's a uniformly fatal disease. And that, by the way, is important here. To even consider accelerated approval, the FDA has given clear guidance. There needs to be one, a treatment of a serious condition with a presumption that this is meeting a completely unmet medical need. Two, that there's a meaningful benefit of drug X over available therapy on a surrogate endpoint, like a biomarker, that is reasonably likely to predict clinical benefit. And then three, this requirement of the study that you just referenced. And so, what we have done is already had extensive conversations with the FDA about such a phase 3 design. And the way we've designed it is we call this study the Restore ALS trial. What Restore stands for is always words in the definition. In this case, randomized is the R, early symptomatic patients is the E and the S, on stable background therapy, which would be riluzole or whatever the doctor prescribes. That's the T. To reduce mortality, that's the R, reduce mortality, and clinical worsening events in ALS. So let's just define that now more clearly. We're going to investigate the effect of CNM-Au8 on improved survival. It's going to be a study planned for 108 weeks. So that's basically 2 years. It'll be double blind. It will be a 2-to-1 randomization with participants on placebo or active drug. And we're literally going to look at, again, survival. It's literally going to have a primary endpoint of time to death or permanently assisted ventilation, which in ALS vernacular means that I'm on a breathing machine because my lungs no longer can power my breath. And that PAV definition means that I have to be on that breathing machine 22 hours of 24 every day, or I will pass away. So that's how this would be
And
designed.
what do you envision the patient criteria being? How, I mean, those are pretty advanced patients, right? When you're talking about being on a ventilator.
Well, again, that's the endpoint is
Right.
we're going to prevent, the goal is to prevent
Okay, gotcha.
ventilator. Okay. So our goal is to see if we could prevent the ventilator and prevent death. And so just
So
to give you,
basically, in a balanced arm, you should have the same number of serious patients versus early patients. And so the events rate should, it hopefully is influenced by the
yeah,
drug. But
that's right. So just to be clear about you're asking about what type of patients would come into the study. And so we're going to let them come on whatever background therapy that they're otherwise prescribed, which in this country is either riluzole or another drug called edaravone or combinations thereof. Those are their only options, really. Most of the world has riluzole. Only a few countries have edaravone. That's it. Okay, so we let them come whatever therapy the doctor wants. Our drug does not interact with other drugs. Our drug is placed together with other drugs without incident. So that's an advantage. And then patients have to come into the study with screening that they have to have symptoms less than 3 years, rather. They have to have already capacity of breathing. They can't be on a ventilator when they start the study. They have to have a certain amount of function. They have to have no sky high biomarkers of neurofilament. In other words, they have ALS, but they're not super late stage, just to make this simple.
sure.
And the goal is we put them on the drug, and the goal is against placebo to prove definitively to the US FDA as a confirmatory study that patients are statistically significantly not dying and not needing the ventilator.
So I
And
just want to clarify, thank you. That's very clear to me. And I want to clarify something else you said. So is it possible after meeting with the FDA in first quarter that they conditionally approve the drug and then set the stage for you to run a confirmatory study? How would that work?
so, yeah, it's a little slightly nuanced difference there, but very important nuance. Before approval, the agency wants to see such phase three confirmatory study underway.
Right.
And
Okay.
they've been very particular about that. In other cases, we're expecting them to be similarly the case in our case. So you asked about timelines earlier. So I mentioned already that all of this biomarker data that we've presented publicly, plus additional that, frankly, still coming in, is being organized into a master dossier of data that is going to be submitted to the agency later this month. And then they'll review that data in the top of the first quarter, and then we'll have a meeting to talk about it face to face in the first quarter. And then what will happen subsequent to that? The goal of this meeting is to review this data in its totality, look at all of the biomarker data from all these different legs of the stool that we've proposed already, and also to look at how that biomarker data is tied to clinical benefit, in our case, survival, to show those linkages, present it to the agency and say, we have a new drug application for accelerated approval, presumptive on a conditional phase three study that would commence. We'd like to submit that for you to you now. Would you please receive its review? And then that will be a review period that would then commence. And then the agency, if they accept all of this, would then give us what's called a PDUFA date, which is a date of potential approval that would happen 6 to 9 months or so, 6 to 10 months later. And so if we do the math and all of that, that would be a decision that would be underway about this time at the end of 26, if the agency is aligned with this planned approach that we are proposing to them. And so the thesis onto the phase three would be that we would also commence and get patients on drug in the year 2026. And we plan to do that as a global effort. So to consider regulatory approval worldwide, not just in the US, we have to enroll worldwide patients. And so we plan to commence this program in Australia, where we have already some deep relationships. We're also working closely with United States sites. And then to get European approval, we would have to do it in Europe as well, Japan as well. There's a number of regulatory markets worldwide that are large enough that they can demand that you have a patient base in our country.
Sure.
So we would do a global study to confirm this. And in the meantime, the agency would consider, do we give people early access, so to speak, through an accelerated approval process, their language, accelerated approval. And that basically gives commercialization access to a drug while the company confirms to the FDA's satisfaction a primary endpoint. So that's
Gotcha.
kind of the vantage point of the next number of months.
So I'm not going to ask you about financing or kind of capital runway. Instead, what I want to talk a little bit about is as an analyst, I kind of believe in the story, and I'd kind of come back to where I started, which is an incredibly clean safety profile, a well-defined mechanism of action in terms of what's happening at a cellular level, metabolic function, survivability, coupled with biomarkers. For us, and for me as an analyst, we look at the risk, probability of success, and the unmet medical need. And that's why we're currently carrying a $23 price target on the stock. Um, we're going to encourage institutions on the buy side to look at what we're seeing. And the reason why I'm a biotech analyst is because it's about the science, right? And it's all about probabilities. And I have to tell you, when I look at the risk reward ratios here, it looks compelling. We are in a tough market environment, but it sounds like you've done a great job in terms of finding non-dilutive capital and kind of keeping things running. And I just wondered, with that kind of introduction, can you wrap things up for me for, the next year and what you see ahead?
So we are super grateful for the support of our shareholder base. We recognize and have recognized from the beginning, as I said, that we are a break with classical convention in our approach. But we also are hopeful that we have a pipeline in a single product here. We've spent all of our time talking about ALS. The same drug has completed three phase two studies in multiple sclerosis. And one of these FDA meetings, of which I referenced, was all about MS. MS is no longer fatal, thank heavens. MS has good therapies presently, but all those therapies do the same thing, tamp down the immune system to prevent the body attacking itself. That's what we're hopeful to have such progress in ALS that we have no longer a survival conversation. MS is no longer fatal as ALS continues to be. However, I raise this because that's what's very innovative about our drug is that this single drug at the same dose is in front of the agency for ALS and in front of a different division in neurology for multiple sclerosis and has Parkinson's work that we plan in the future, all the same drug, same dose, because the mechanism of how we are driving energetic metabolite support to a system under compromise is the same. So, back to your point, we've had a number of investors that over the years have understood this, have concurred with our vision such that they've parted with their cash, not only when we were private during, the period I joined in 2013. We took the company public 7 years later in late 2020. We've been public since then. And so we have been grateful both in our private sphere and our public sphere as a NASDAQ listed company to have the support of investors. Now, we've seen a lot of up and downs with the stock market. The biotech market is like that, and Clene in particular has been. But your investor viewers could recognize that if in fact our drug is approved, if the FDA decides to let us file a new drug application, and these are all ifs, we have to wait and see what 26 will give us. But if the drug is approved and we could start selling, then Clene is no longer its present market cap. Clene would be a larger market cap. In fact, all three ALS drugs that launched when Amylyx launched another drug that was temporarily in the space, they all launched at about the same price. Uh, there's been a lot there's a lot of opportunity in ALS for a drug that is approved. And we see this right now with our compassionate use EAP programs. We could give as much drug away as we could possibly manufacture. In fact, we desperately wish we could provide for free more drug than we presently do. 500 patients and counting is kind of our cap. It's an extraordinary effort. But anyway, back to your point, is that over 2026, we appreciate your understanding and support, Jason, but over
Thank you.
2026, the opportunity is that if we can get this drug in front of the agency and if they would consider our approvable path such that we can get approved, then it's a great opportunity for investors, very different from the present price, as you can appreciate.
Rob, I look forward to checking in with you, post first quarter after you've had that FDA meeting, because it strikes me that just there's going to be a lot of activity in the next 12 months. Very exciting. You tell the story well. Thank you so much for sharing your time. Anyone interested in seeing our model or seeing the reports, just go to our website and you can pull it down. Rob, thanks so much. Have
Thank
a
you.
great holiday.
You too, Jason. Thanks so
Thank
much.
you. Bye bye.